Okay, ladies and gentlemen. Well, when we left off last time, we were looking at mutation rates in DNA. And this time, I'd like to begin by looking at a specific type of mutation, gene duplications. Now, gene duplications are really important in evolution because they basically create raw material for future change. When the replication machinery messes up or through other means genes are copied, you create more basically genetic material that can be exposed to other mutagenic factors down the line, or can experience mutation down the line. So you create basically more and more material, and in this additional material you have more opportunity for new genes to be created. So gene duplications, again, are critical evolutionarily because they lead to the creation of more genetic material and then ultimately help increase the raw material needed for evolutionary change. Now, there are two basic types of duplications that occur. One is through the creation of what's called orthologous genes. Now, this happens when genes are duplicated or homologous genes are duplicated in separate species. So you have this sort of ancestral gene here, and then when speciation occurs, that ancestral gene is passed on to each of the different species, but then mutations can occur, so you have related but slightly different copies of the gene in different species. Paralogous genes, on the other hand, are duplications within the same species. So you can create multiple copies of a particular gene within an individual species. And the example the book cites is the family of genes, or the set of genes related to our olfactory senses, our ability to smell. We know that on these receptors, we have, on our olfactory receptors, we have very specific proteins that can detect molecules of specific shape. Well, if we have all these copies of these paralogous genes created and various mutations occur over time, well, then you have lots of slightly different receptors that allow us to detect different scents and aromas over time. So again, gene duplication provides raw material for the creation of basically new traits. So an important point. All right. Now, this is a critical idea. This is guaranteed AP material. It's the idea of relatedness as identified through endosymbiosis. Endosymbiosis is a very important idea. Lynn Margulis pioneered the idea. I think it was back in the 80s, late 70s, early 80s. She was looking at this endosymbiotic theory. The basic idea is in ancestral species long ago, cells invaded one another, and rather than killing each other off, formed this symbiosis, this mutualistic symbiosis. And over time, through evolution, you create these new organisms that are a combination of ancestor species. So it's believed that a bacterial cell invaded another bacterial cell long ago, and as a result of that, connection was made in which cellular respiration occurred. And obviously there are great benefits to that, being able to take in oxygen and use that as well as sugars to produce ATP. Now, after that, there was also the opportunity for photosynthesis to occur. And again, huge benefits to that, because now you have an organism that can make sugars and break down sugars. Now, looking at the DNA that's found in chloroplasts and mitochondria, again, they have their own DNA, they reproduce in their own cycles. We see that the DNA in our mitochondria and in the chloroplasts of plants is more closely related to bacterial cells than to eukaryotic cells. So it's a strong connection that we have with bacteria, and strong genetic evidence of evolution. Because again, we find that our mitochondrial DNA and the DNA in chloroplasts is bacterial in nature. So that's pretty cool. All right, so how do we organize all this stuff? How do we organize all these different forms of life? And we do that through cladistics. Now, clade is basically a group, okay? So it looks at how can we put together these different groups to get a sense of evolutionary history. So again, we group these organisms into cladograms based on shared characteristics and then unique characteristics. The whole idea is to begin with this common ancestor that has a whole host of characteristics that will be shared by other members of the group. And again, we work to look at increasingly exclusive characteristics that make us able to point out individual organisms. So in terms of how you make a cladogram, you look at various traits like you see here in the chart, and then figure out how many species have particular traits. You denote a 1 if they have the trait and a 0 if they don't. Now doing this, you group them so that everyone has the most characteristics in common. So we start with a vertebral column, okay? And then they all have a vertebral column, and then you move on to the idea of hinged jaws. Okay, how many of these organisms have the hinged jaws? We'll make this the hinged jaw group since there are these 4 left. And then, okay, of these 4, who has walking legs? These 3. And then you come up with these increasingly exclusive groups. And again, that shows us an idea of when these different traits occurred. Well, what evolved first, a vertebral column or hair? Well, based on our cladogram, it looks like hair evolved after a vertebral column. Now, an important characteristic in setting up a cladogram is identifying what's called an outgroup. This is a group that has evolutionary history, okay? So a lancelet has a notochord, although it doesn't have a vertebral column. So it is a chordate, but it doesn't have a vertebral column or any of these other features that these other organisms have. So it basically sets up our connection with an ancestor species. These different branch points identify common ancestors, and then you have these branches that occur and identify individual organisms. All right, now certainly you can say, oh my gosh, there are all these characteristics. How are we going to know how organisms are related to one another? There are a couple of rules of thumb to work with here. One is by looking at the rule of maximum parsimony. The whole idea is let's just try and give the most straightforward, most simple answer. You need this sort of simple explanation for relationships, and that's your first goal. So you start with what's the simplest way to organize all these organisms, and then we have to look at this rule of maximum likelihood. So again, the idea is the fewer differences there are between organisms, it's probably more likely the more closely related they are. So you look at relationships based on how similar their DNA is. Now, we can apply this to try and classify organisms in different ways. You can look at these different species of birds. You think, oh my gosh, they have some characteristics in common. Look at beak shape, but coloration, the structure of the wings and tail feathers, it might differ. So how are we going to create a phylogenetic tree that best predicts their relation? Well, what you do is look at their similarities. Okay, so you look at this DNA sequence. Here's our outgroup, our ancestral sequence, and then you just look at how many bases do they have in common with one another. Okay, so you run through the different possibilities, and again, you use computer software to do this, but you try to come up with a straightforward answer that has minimal number of differences, and then that is generally what we look at for a prediction. Okay? Now, when you do this, you're going to come up with different types of groups, and there are 3 basic types of groups. I know there will be quiz and test questions related to this, so please make sure you're comfortable here. One group is called a monophyletic group, mono meaning one, phylum, again, this sort of family history. A monophyletic group has a single ancestor and then all that ancestor's descendants. Para, meaning beside or around, paraphyletic groups have a common ancestor and some of the descendants, but in this case not all. Here G is left out. And the final group that we'll look at is a polyphyletic groups, many different phyla or families, if you want to think of it that way. So what you do is look at descendants of more than one common ancestor. So we have different common ancestors and some of the groups of their descendants here. Now, what we want to do is try and get it set up so that we have these individual monophyletic groups. If you do that, you start with the features shared by all of the groups, and then you get to these, again, increasingly exclusive characteristics. So again, the idea is to create up sort of nests of all these monophyletic groups, and then that will give you a sense of relatedness and when different traits evolved. Okay. So again, we can just go back and look at relationships between DNA and get a sense of relatedness over time. So again, we look at all these different features. We know that morphology isn't always the best way to go, so we have techniques for looking at relationships between DNA and species, and it allows us to answer really important questions like, what is this organism more closely related to, lizards or snakes? And here you have your answer. Okay? Scary no matter what, right? All right, I hope everyone does well, and look forward to seeing you all in class soon. Ta-da!